Imagine a piece of software, right? Like a massive program with 2.8 billion lines of code.
That's a lot of code.
Yeah, exactly. Now imagine going into that massive system, finding one specific line and just changing a single letter, like just a tiny microscopic typo.
Just one keystroke.
Right. And because of that one single keystroke, the entire system completely reverses its core output.
It's wild to think about.
It really is. And today we're actually looking at how that exact scenario plays out inside human DNA, like how a single genetic typo can completely reverse human biological sex. So welcome to today's deep dive.
Glad to be here. We've got a really fascinating one today.
We do. We are pulling from this incredible article in Psychology Today by Dr. R. Douglas Fields. And he unpacks this recent, honestly, paradigm-shifting study published in Nature Communications by lead researcher Nitsan Gonin. She's over at Baralon University.
Right, and the research is just groundbreaking.
It is. Our mission today, for you, the listener, is to figure out the mechanics of this genetic reversal. And, we also need to understand why the medical establishment has basically spent decades looking in the completely wrong place to understand differences in human sex development.
Yeah, to really grasp the sheer scale of what Gonin's team discovered, we kind of have to talk about the physical landscape where this mutation actually happens.
The environment itself.
Exactly, because for the better part of 50 years, modern genetics has been operating with this massive blind spot regarding our own biological code.
Which is crazy to think about now.
Oh, totally. Like when the Human Genome Project really kicked into gear back in the 1990s, the primary obsession was just finding genes. The whole scientific community was, hunting for the specific sequences of DNA that actively produce proteins.
Because the proteins are the things that actually build the physical structures of the body.
Right. They're the bricks and mortar.
But the Psychology Today article highlights this statistic that, it really puts that whole gene hunting obsession into perspective. Because 98% of our DNA does not actually code for genes.
Yeah, 98%. It is a staggering ratio.
That's almost all of it.
I know. And because that 98% didn't actively manufacture proteins, early geneticists basically just dismissed it.
They just ignored it.
Pretty much. They labeled this massive expanse of our biology as junk DNA. Like they literally assumed it was just evolutionary static or leftover baggage accumulated over millions of years.
The arrogance of looking at 98% of our own instruction manual and just assuming it's useless filler, that is wild.
It's definitely a product of its time. But Dr. Fields makes it clear that this perspective has entirely shifted now.
Right.
We now understand that this non-coding region is essentially the routing software of the body. It regulates whether a specific gene is activated to produce a protein or whether it just remains dormant.
Okay, so it's the ultimate control layer.
Exactly. Because here is a baseline reality of mammalian biology. Every single cell in the human body contains the exact same hardware.
The identical genetic blueprint.
Yes. The blueprint to make every single protein is just sitting there in every cell. The DNA coiled inside a brain cell is it's indistinguishable from the DNA coiled inside a bone cell.
Okay, wait, hold on. Let me just unpack this. If my bone cells have the complete brain cell manual, then the only difference between my skeleton and my nervous system is just like which pages of the manual are glued shut.
That is the biological reality.
Wow.
Yeah, and the mechanism gluing those pages shut or, forcing them open is that 98% of non-coding DNA. It acts as a routing manager.
Directing traffic.
Right, ensuring that only a highly targeted subset of genes is executed in any particular cell, while the rest of the genetic hardware is strictly suppressed.
Okay, because the mechanics here are what really matter. And I'm stuck on this idea of the routing manager. If this 98% of our DNA doesn't actually build any physical proteins itself, if it's just directing traffic like you said, how does a single typo in that routing code physically build an entirely different reproductive system?
Well, to answer that, we have to look at the chain of command. We have to zoom way down to a highly specific microscopic mechanism. Gonin's research focuses on a non-coding sequence of DNA known as a regulatory element.
Okay.
Specifically one called N813.
So N813 is like a specific line of code within that 98% routing software.
Correct. And to understand how it functions, we really have to move past those old middle school biology metaphors. N13 isn't a physical padlock. It operates much more like an authentication token in a software API.
Oh, interesting. So how does that work in the cell?
So floating around inside the nucleus of our cells are these complex proteins called transcription factors. You can think of a transcription factor as a login request.
Okay, a login request.
Right. When it physically encounters the N813 sequence on the DNA strand, it tries to bind to it.
And if the chemical passcode matches, the system grants access.
Exactly. The transcription factor has to fold in three-dimensional space to perfectly match the biochemical signature of Na13.
Like a hyper-specific puzzle piece.
Yeah. And if the sequence is correct, the transcription factor binds, the authentication clears, and Na13 executes a massive command and control sequence.
Which means it reaches across the genome to grant read and write access to the exact genes required to form reproductive organs.
We got it.
That requires just an unbelievable level of precision, because I think we tend to visualize the inside of a cell as this chaotic, fluid biological soup.
Most people do, yeah.
But it's actually an incredibly rigid, highly engineered authentication protocol. I mean, the transcription factor is navigating a nuclear soup containing 2.8 billion base pairs, looking for a sequence of maybe a dozen letters that match its specific access request.
Right. And because the authentication protocol is so precise, it is intensely vulnerable to disruption.
A single typo.
Exactly. If the sequence is altered, the transcription factor either can't bind or the wrong transcription factor binds. The routing software then executes a completely different command.
Which brings us to the core mechanism of the Nature Communications study. And, this is where the logic of sex development completely flips on its head.
It really does.
The researchers took XX mice, which, just as a reminder, carry the chromosomal hardware to develop female reproductive organs.
Right. XX is typically female.
And they went into that 98% non-coding region. They targeted the NAS13 regulatory element, and they introduced a single-letter mutation.
They changed a single base pair in the DNA code.
And by changing that one letter, those XX mice developed testicles and male genitalia.
Yeah, it's astonishing.
But let's break down the actual biological domino effect here. How does breaking a piece of code in a female mouse actively build male anatomy?
This gets right to the heart of how biological sex actually developed in mammals. The baseline developmental pathway, like the default physical architecture, is female.
Okay, the default is female.
Right. To diverge from that and develop male reproductive organs, the biological system has to actively deploy a massive intervention. It has to turn on a highly specific set of genes, most notably a gene called SOX9.
SOX9.
Yes, and SOX9 overrides the default female pathway and initiates male development.
So the body basically has to actively hit a male override switch.
Yes, that's a great way to put it. And NH13 is the authentication token that authorizes that override.
Okay, I'm following.
In XY males, a transcription factor binds to NH13, activates it, and SOX9 gets flooded into the system, building male organs. But in XX females, that exact same NH13 sequence is just sitting there in the genome. It still exists.
It's because we all have the same blueprint.
Exactly. So, for proper female development to occur, NH13 must be heavily repressed. The biological system has to actively block that token from authenticating, ensuring SOX9 stays turned off so the default female pathway can proceed.
Okay, here's where it gets really interesting for you listening. Because the mutation the researchers introduced in the XX mice, it didn't randomly invent a male reproductive system.
No, not at all.
The typo they introduced specifically broke the repressor binding site on NN13.
It destroyed the security block.
Wow. So the system lost its ability to suppress the male override switch. Even though these mice had XX chromosomes, that single typo allowed the authentication token to bypass security.
Right. N13 just ran wild. It triggered the male genes and completely reversed the biological sex of the organism.
That is just, it's mind-blowing.
It is the ultimate proof that the chromosomes are not the sole dictators of sex. And Ghana's team proved this is a two-way street, by the way.
Oh, right, the 2024 study.
Yes, in 2024, they ran a corresponding study looking at XY mice, which normally develop into males. They introduced a different microscopic mutation into NN13, but this time they broke the activator binding site.
So they broke the mechanism that triggers the male override.
And the exact opposite outcome occurred. Because the activator was broken, NN13 couldn't authenticate the override.
So SOX9 never turned on.
Exactly. The SOX9 genes were never activated, and despite having a Y chromosome, those mice developed entirely as females.
That is incredible. The author of the article actually quotes Gonin describing NA13 as a double-edged sword. And that mechanism just makes perfect sense now.
It really does.
It's a single, incredibly fragile node of routing software that holds the power to push physical development in two completely opposing directions, entirely independent of the underlying chromosomal hardware.
Yeah, the physical reality of the chromosomes was entirely overridden by a single typo in the non-coding space.
Yeah.
And we really need to bridge this from laboratory mice to human reality.
Because we share this exact same regulatory architecture, right?
We do. The E13 region exists in the human genome, and it regulates our development through the exact same authentication protocols.
And the human implications of that are massive. Dr. Fields notes in the article that roughly 1 in 4,000 newborn babies presents with differences in sex development, which is commonly referred to as DSD.
Right.
So you have thousands of families navigating this incredibly complex medical reality, seeking answers about their child's biology.
But the medical establishment has, unfortunately, historically, failed to provide those answers. And it's largely because of how genetic diagnostics are currently structured.
The blind spot we talked about earlier.
Exactly. When a patient with differences in sex development goes in for genetic testing, hospitals almost exclusively use a diagnostic tool called whole exome sequencing.
The exome being the 2% of our DNA that actually codes for genes.
Correct. Whole exome sequencing is faster, it is significantly cheaper, and it perfectly aligns with that decades-old bias that only the protein-coding genes really matter.
Right.
So the geneticists scan the exome looking for a broken gene, like a broken piece of hardware, that explains the biological development.
But they are completely ignoring the 98% routing software.
They are completely ignoring it. Because N13 exists in the non-coding region, the so-called junk DNA, this vital sex-determining switch is entirely invisible to standard medical testing.
That's incredibly frustrating.
It is. Doctors are spending months analyzing the bricks, completely blind to the fact that the project manager's clipboard has a massive typo on it. It is an institutional diagnostic blind spot.
Man, if the medical community has been completely missing this switch, it really makes you wonder how many people are walking around with a biological reality that directly contradicts the simple labels we use.
Oh, undoubtedly.
And Dr. Fields actually brings up a really fascinating point at the end of his article about how this changes our cultural conversations. And, we always want to be super clear with you all here. We aren't taking a political side here. Our goal is strictly to share the ideas presented in the original article.
Absolutely.
But the author notes that against the backdrop of current government controversies and vitriol regarding transgender people, these findings provide a lot of food for thought. It proves that biological sex isn't a clean chromosomal binary. It is this vast spectrum governed by microscopic, fragile mechanisms.
It is a critical scientific reality that pushes back against the rigid categorizations society often tries to enforce.
Yeah.
When the very foundation of biological sex development hinges on a single microscopic authentication protocol, hidden in a region of DNA we used to casually dismiss as junk, the idea that biology is a simple black-and-white coin flip just becomes scientifically insupportable.
It requires a fundamental shift in how we understand our own blueprints, because the complexity is the point.
The complexity is entirely the point. And Gonan emphasizes that the discovery of N13's mechanism is likely just the beginning of understanding that complexity.
Oh, really?
Yeah, she notes that this specific regulatory element is merely the tip of the iceberg when it comes to the non-coding DNA required for sex development.
Which means there is an entirely uncharted universe of routing software dictating our physical reality that we haven't even begun to map.
There are likely hundreds of other regulatory nodes, authentication protocols, and repressors hidden in that 98%, all working in tandem to regulate development.
Just waiting to be found. Exactly. The lab at Baralon University is actively working to systematically identify these elements, which could eventually revolutionize whole genome sequencing and, finally provide accurate diagnostics for these patients.
So what does this all mean for you listening? Let's recap the critical shifts in understanding we've explored today. First, those X and Y chromosomes are not the ultimate definitive dictators of biological sex that we assumed they were.
Right.
Second, the 98% of your DNA that the scientific community confidently dismissed as evolutionary junk is actually the master software regulating your entire physical architecture.
And third, the margin between developmental pathways is staggeringly thin. A single one-letter typo in a non-coding authentication sequence can completely override your chromosomal hardware. It can break the repressor systems and completely reverse biological sex.
It really completely dismantles the neat, orderly biology textbook we all grew up with.
It does. It forces us to acknowledge that the human body is a dynamic, continuously regulated system rather than just a static set of genetic building blocks. And it leaves us with an incredibly profound question to consider. If researchers are calling this just the tip of the iceberg, and a single letter in our non-coding DNA can reverse something as fundamental as biological sex.
Whoa, what other massive life-altering traits are sitting quietly in the remaining 98% of your junk DNA, just waiting to be discovered?
Right. What else is in there?
It is entirely possible that the answers to our most complex physical and neurological traits are hidden in the exact places we decided weren't worth looking at.
A wild thought to walk away with. Thank you for joining us on this deep dive into the dark matter of the genome. The next time you find yourself assuming that the basic rules of human biology are settled science, remember just how much of the manual we haven't even bothered to read yet. Keep questioning the code.